So let's talk a little bit about the discovery of the double helix. It was discovered by a couple of scientists. Well, two scientists got the Nobel Prize for the discovery of the DNA double helix, and that was Watson and Crick. James Watson and Francis Crick. And these were two guys that didn't actually like each other all that much, though they collaborated on this research project. And really what they did was they took information that others had, this is just the process of science, that others had generated, saying that yes, it is in fact DNA that conveys information. And they compiled a bunch of that information, and they came up with this helical structure. Now, another individual that contributed to the discovery of the DNA molecule was Rosalind Franklin. And Rosalind Franklin used a process called X-ray crystallography. And X-ray crystallography basically takes X-rays and blasts it at molecules, and then those X-rays are deflected off to some absorbent surface in this chamber that's holding that molecule that you're blasting the X-rays at. And what you can do with that reflection or deflection pattern is you can reconstruct the three-dimensional conformation of a molecule. Watson and Crick didn't know how to do that. Rosalind Franklin did. So she did it, and they were able to take her images and generate that structure. Now, as I said, only Watson and Crick were given the Nobel Prize for it, which seems incredibly unfortunate for Rosalind Franklin, right? One of the reasons that people suggest that she didn't receive part of the Nobel Prize was because she was a she. Now, the Nobel Prize can't be awarded posthumously. That is, you need to be alive to receive the Nobel Prize. And when it was awarded to Watson and Crick, she had already passed on. And so it could have been that had she been alive, she would have also received it. But there are the skeptics, of course, that suggest that it was delayed in awarding it until she already passed. Anyways, the bonding always occurs between A's and T's and G's and C's. We have two classes, right, of nucleotides, purines and the pyrimidines. Purines are always going to be A's and G's, regardless of whether it's DNA or RNA. And the pyrimidines are going to be C and T if it's DNA, or C and U if it's RNA. Okay, now, again, the important function of genetic material, of DNA, is to communicate information, right? The way that we use that is twofold. The first of which is to pass information about ourselves to our offspring. Secondly, the DNA molecule is constantly used as a blueprint for the construction and manufacture of proteins. All right, are there questions about that? Questions about?
Can you say what you just said again?
Sure. The function of nucleic acids, DNA specifically in our body, is twofold. The first of which is to pass information to our offspring, right? The second of which is the DNA molecule is constantly being used as a blueprint for the manufacture of proteins. Okay. You bet. Other questions about nucleic acids? So I want to shift our attention really quickly to chapter 4.